Welcome back to Biology 2225L, Anatomy and Physiology Lab at CNM. The topic this week is male reproductive system, and most of the concepts can be found in chapter 27 of your textbooks. Now, before I start talking specifically about the male reproductive system, I want to introduce a couple of ideas regarding reproduction and sex in general. These are not on our list of terms, and these slides are not in the PowerPoint that I provided for you. I just added a few introductory slides to the PowerPoint. Again, because I think it's important to introduce the basic concepts of reproduction and sex first, and then move on to talk about the male and then next week female reproductive systems. So we'll begin with the concept of reproduction in general. Reproduction is the increase in the number of individuals in a population of organisms, which is a fancy way of saying it's making babies. Sex is often confused with the term reproduction, but it actually is something different. Sex is a genetic process whereby genetic material from more than a single source is combined together into one organism. So it's the bringing together of genetic material from more than one individual into a single individual. And we often think of these two things as being synonymous, but in the living world, there's actually lots of variation around these themes, and it is possible for some organisms to reproduce without sex, and it's possible for some organisms to have sex without reproducing. So in other words, uniparental sex, sex involving only a single parent, would be obviously reproduction, making babies, but you only have a single source of genetic material, the single parent. Binary fission, parthenogenesis, there are various examples of reproduction involving only a single parent. Perhaps one of the more common examples here in New Mexico is the New Mexican whiptail lizard. It's a striped lizard with a long blue tail that most of us have seen wandering around even at the CNM Westside campus. This is the state lizard of New Mexico, by the way. This also happens to be an all-female species. There are no males in this particular species. It's all females, and the females simply lay eggs without needing males to fertilize them, and the eggs essentially hatch, and the offspring are genetic copies, clones essentially of the mother. So a single source of genetic material, that would be reproduction without sex. Sex without reproduction is less common, but it does occur in certain organisms. Paramecia, for instance, a single-celled organism, two paramecia can come together and exchange what are called paranuclei, which are tiny bits or fragments of their DNA, exchange that with one another, and then go on their separate ways. And so they have come together and mixed genetic material from more than a single source. Each individual now contains a new mixture of DNA, but no reproduction will have occurred. We started with two organisms and we ended with two organisms. So it is possible for these two phenomena to be distinct, and they are distinct in the natural world. So again, what that means is reproduction can be either sexual or asexual. If you think about it from an evolutionary perspective, there are advantages to asexual reproduction. It is quick and efficient, meaning you don't have to spend time looking for a mate. You don't have to spend time attracting a mate, doing any kind of mating rituals or dances. And it's efficient in the sense of genetically, asexual reproduction means you are 100% related to your offspring as a parent. And so in terms of passing genes on to the next generation, which is how we measure fitness of organisms, it is 100% efficient compared to sexual reproduction, which, as we'll see, is only half as efficient. The drawback of asexual reproduction is that over the long run, it doesn't generate much genetic diversity, because generally speaking, it doesn't involve the mixture of genetic material from multiple sources, or at least two sources, the offspring that you produce are generally more genetically homogeneous. And in some cases, they're all identical, like the whiptail lizard that I mentioned earlier. The daughters are all essentially genetically identical to their mother, and so they're also genetically identical to each other. And so that's a species with very little genetic variation among its individuals. The only source of variation there would be mutation, spontaneous mutation. Sexual reproduction, on the other hand, requires two parents. Interestingly enough, we never see a situation where there's more than two sexes involved in sexual reproduction. Theoretically, it's possible, but practically speaking, two sexes seems to be the most economical way to have sexual reproduction. Its drawbacks, again, are that it's time-consuming and costly. It can be very difficult in some species to just locate a mate, and then mating rituals, etc., consume time and energy. Its biggest drawback, of course, is, again, offspring are only 50% related to each of the parents, and so right off the bat, as a strategy, it's 50% less fit than asexual reproduction. Yet, sexual reproduction is very common, and probably most common among organisms that we're familiar with anyway as humans. And so it has to have some benefit to offset these costs, and the benefit is that the offspring are genetically diverse. And in time, of course, the environment is bound to change, and sexual reproduction produces a genetic diversity of offspring. And so it's like the old adage, don't put all your eggs in one basket. Don't put all your genes in one kind of offspring, right? Produce a diversity of offspring, and when the environment changes, you have an increased likelihood of having at least one offspring who's genetically suited to a new environment. Now, we used to think about this in the context of environmental change, things like global warming, that were relatively slow changes traditionally, and you wouldn't think you would need sexual reproduction every generation to deal with these non-living or abiotic factors that tended to change relatively slowly. So to justify why organisms become obligately sexual, meaning reproduce sexually all the time without any asexual reproduction, you'd have to find something in the environment that changes so quickly, so rapidly, that you would need to produce diverse offspring every generation. Well, what biology settled on as the kind of environmental factor that changes that frequently, requiring so many organisms to be sexual all the time, is pathogens and parasites, disease-causing organisms. We know that because of their short generation times, meaning how quickly they produce new generations, bacteria, viruses, things like that, they evolve at a much faster rate than do, generally speaking, the hosts upon which they live. And given that bacteria and other pathogens are constantly adapting to the body of the host, it really is incumbent upon the host to be sexual in most cases, so that all of their offspring aren't born automatically susceptible to whatever pathogenic organisms have already adapted to the parent. And this is what we think really ultimately justifies why so many organisms are sexually reproducing organisms. And we can certainly appreciate the value of that diversity when we face something like a pandemic like COVID-19. We see in the population of humans there is great variation, genetic variation, between individuals who are asymptomatic when they contract the virus to people who, of course, pass away when they contract the virus. This is a great example for us of the genetic diversity that exists in our population, primarily due to the effects of sexual reproduction. If we were an asexual species and we happen to be an individual or come from an individual that was harmed by COVID-19, it's possible that the virus would have severe effects on every single individual in the population. And so there you can see the drawback of asexual reproduction in a population. It just makes it more susceptible. to something like COVID-19. With sexually reproducing organisms, there's usually some individuals in the population that will be resistant, and the species will continue. So as we said, sexual reproduction requires two parents, two sources of genetic material, but it's also interesting to note that it doesn't always require two sexes. And so that raises, of course, the question, how can you be a sexual species and yet not have two sexes, male and female? Well, the terms male and female actually do have a biological definition. The biological definition of male is the organism that produces the smaller motile gamete. A gamete is a reproductive cell that is involved in sexual reproduction. The key feature of a gamete is that its number of chromosomes, its genetic content, is half the normal content. This way, when you take two gametes and bring them together in a process called fertilization, the chromosome number or genetic content adds back up to the normal number of chromosomes. So gametes, again, are reproductive cells specifically designed for sexual reproduction that carry half of the normal amount of genetic material. So males produce gametes that are relatively small and motile, meaning capable of movement, independent movement. Of course, what we're talking about here are sperm cells for humans. Female is defined as the organism that produces the larger sessile gamete, meaning compared to the male gamete, the female gamete is larger, and it is incapable of independent movement. Now, it might get swept along by currents or the environment, but on its own is not capable of independent movement. And this, in humans, is what we call the ovum. So, in biology, male and female have a very specific meaning at an ultimate level anyway. Now, in terms of expression of male and female characteristics, there is some variability there in terms of what we call the phenotype, or the actual characteristics of an organism. But at its foundation, going back to first causes, male and female do have an ultimate definition in the shape of the gametes. So how do you get sexual reproduction in organisms that don't have sexes? Well, some organisms are what we call isogamous, which is a fancy way of saying that all the gametes that are produced by all the individuals are structurally similar. There is no difference. There are no two different kinds of gametes. You don't have one that's smaller and motile and one that's larger and sessile. They're all the same. They might all be, essentially, they're all the same size, and they may or may not be capable of independent motion. So in the absence of discrete or different kinds of gametes, you, of course, then don't have discrete kinds of organisms. You don't have male and female. So there is sexual reproduction because gametes still fuse together to form offspring. There's just no male or female. Now, in some cases, there are mating types. There are biochemical differences between the types of gametes produced, and one gamete would have to be one biochemical type in order to fertilize a different biochemical type, but that's nothing you can see. That's not a physical trait that you can see. So sometimes we'll have isogamous organisms that have mating types. Mating type positive, for instance, has to mate with mating type negative, but we can't call that male and female because the gametes do not differ in size. Other considerations that I don't have here, but just something to think about in terms of reproduction and sex, is once fertilization does take place and a zygote is produced, that's what we call the first cell produced by the fusion of two gametes. It's called a zygote. Once that happens, we have some other things to think about. For instance, where did that fertilization take place? If the fertilization of the egg takes place out in the environment, that's referred to as external fertilization. This is very common in many organisms, and in vertebrates, the the organisms that are closest to us, we see external fertilization in things like fish and amphibians, where simply the female lays her eggs in the water and the male swims over the egg mass and releases his sperm into the water, and the sperm swim through the water onto the egg mass and fertilize them outside of the female's body. That's external fertilization. It's somewhat inefficient because water currents can sweep away sperm cells, and only a few might actually make it to the egg mass. It also gives the female less control over who fertilizes her eggs, because other males can swim in at the right time, of course, and release their sperm onto the egg mass. If you want more control and more efficiency, you go with what's called internal fertilization. With internal fertilization, the sperm cells are delivered into the body of the female, and the fertilization takes place inside the body of the female. This is seen in things like reptiles, birds, and mammals, for instance. And so, of course, humans being mammals, we exhibit internal fertilization, where the sperm is delivered into the female body. Again, this is more efficient because you don't have to produce as much sperm, and it gives the female more control over exactly which male is going to fertilize her eggs. And then a final distinction that we can think about is, where is the development of the zygote going to take place? The zygote, of course, begins to undergo cell division and very rapidly becomes an embryo, and then after about 8 to 9 weeks, the embryo becomes what we call a fetus, and it continues to develop. Where that development takes place can vary amongst organisms. So, of course, if fertilization was external, then development is going to continue to be external. But even with organisms that have internal fertilization, most of the development might be external. And what I mean by this are, of course, the organisms that are egg-laying organisms. And so, generally speaking, as I said, reptiles and birds have internal fertilization, and in most of them they lay eggs, and most of the development of the embryo/fetus takes place outside of the female's body. That's external development. There are some reptiles that have live birth, but this is not the same as the kind of live birth we see in things like mammals, because in most cases, essentially what they have is they have eggs that they just don't lay. The eggs are retained inside the female's body, and the eggs hatch inside the female's body, and then the young pass out of the female's body. So it may look like they have a placenta-like relationship with the offspring, but they really don't. It's just that the eggs are not laid. So that's kind of an in-betweener between internal and external development. Mammals are the organisms that have taken internal development to its highest heights, in which we see that instead of laying eggs, most mammals retain the embryo/fetus inside the female body, and the female takes great care of the fetus, nourishing it and protecting it through the entire term of its development. Now, I said most mammals because there are 2 mammals that lay eggs, and this surprises many people. You would think live birth is one of the basic fundamental characteristics of all mammals, but it's not, because there are 2 mammals that do in fact lay eggs and exhibit external development, and that is, these are both very ancestral kinds of mammals. They're called monotremes. They're in the order monotremes. They live in Australia and New Zealand, and they are the platypus, which most people have heard of, duck-billed platypus, and something fewer people have heard of, something called an echidna, which is very much like a hedgehog type creature. But it is true that most mammals, other than those 2, exhibit internal development where we take great care of the developing offspring. The hallmarks of being a mammal, by the way, are not live birth, but hair and milk production. The production of milk to feed offspring is what makes mammals different from all other organisms. So as humans, we are generally speaking sexually reproducing organisms that perform internal fertilization and internal development. And so with that all being said, as we look at the male and female reproductive systems, we have to keep in mind that they are designed for those particular purposes. They are designed to allow for internal fertilization and internal development. Okay, so at this point it might be interesting to ask, are there any examples of asexual reproduction in humans? Does it ever happen naturally? And the answer is yes, it does. Technically, it does. And the circumstance where humans reproduce asexually would be in the case of identical twins. If you think about what happens in identical twins, you actually begin, the organism begins as a single zygote, developing in the female uterus. And that zygote will start dividing to become an embryo, 2-cell stage, 4-cell stage, 8-cell stage, so on and so forth. And at that point, it's a single organism. It is a unique, novel, genetically novel, new organism. But something can happen in some cases early in development where that early mass of cells, which is still technically a unique organism, splits in half. Actually splits in half. If this happens early enough, then each mass can develop into an entire human being. And this is exactly what happens in identical twins. So we start with a single zygote and a single mass of cells, that's a single organism, and we end up with two masses of cells, which become two organisms. So we've had reproduction, we've had an increase in the number of individuals, but that split didn't involve the combining of DNA from more than a single source. The two masses came from a single source, and so technically the splitting of a mass of cells to produce identical twins within the uterus of a mother, technically that's an asexual reproduction event that occurs very early in the life of that particular organism. Sometimes the split doesn't occur all the way, and we end up with what are called conjoined twins, where basically you have identical twins that are still, to varying degrees, maybe still attached to one another. In some cases, they're so attached that they share vital organs and can't be separated. In other cases, they're attached only slightly, and it is possible to separate them, and they become two separate individuals. Of course, as is often the case with biology and nature, solid boundaries are sometimes difficult to find, and what constitutes two separate individuals, two unique individuals versus one individual kind of gets blurred when you talk about conjoined twins. All right, so as we return to the PowerPoint that the authors of your book provided, you see that they talk about some of the things that I've just talked about, though they don't go into the detail that I just went into. So again, sexual reproduction is biparental. Genetic material comes from two parents, and it is sexual reproduction. There's genetic diversity among the offspring. The gametes are the sex cells that come together during fertilization. A gamete comes from each parent, and remember, the gametes again have half the normal number of chromosomes that a typical somatic or body cell would have. The zygote, once again, is the new individual, the first cell of the new individual, and it is the fertilized egg that has a combination of both father and mother's genes. Again, the male and female gametes are referred to as sperm and egg. And, as we see here, the definition of male and female really is defined by what kind of gamete you produce. So if you produce the smaller motile gamete, again, the sperm, then you're male. If you produce the larger sessile gamete, the egg, then you are considered to be female. Now, what determines whether an organism is going to make sperm cells or egg cells is also something that is not the same for all organisms. In humans, whether you're male or female is determined by what are called sex chromosomes. And so remember, we have 46 chromosomes altogether, and they exist in pairs called homologous pairs. So 46 chromosomes means we have 23 pairs of homologous chromosomes. 22 of those 23 pairs are truly homologous chromosomes that carry genes for the same traits. But one pair of the 23 pairs is what we call the sex chromosome pair. And recall that there are actually not homologous chromosomes in the sex chromosome pair, but we have two potentially different kinds of chromosomes, the X chromosome and the Y chromosome. And this is what we learned in intro bio when we talked about basically Mendelian inheritance and sex-linked traits. In humans, females are the organisms that carry two X chromosomes and do not have a Y chromosome. In males, in humans I should say, males carry a single X chromosome and a single Y chromosome. And it's this difference in the kinds of chromosomes that are found in the cells that determine ultimately whether in humans a organism is going to develop into a male or female. This is not always the way that sex is determined. In birds, it's often the opposite, that females are the ones that have two different kinds of sex chromosomes and males are the ones that have two that are the same. And sex isn't even necessarily always determined genetically. So alligators and crocodiles, for instance, are famous for having temperature determination of sex. Literally, the sex of a crocodile or an alligator is determined by the temperature of the nest in which the egg develops. And so if the temperature is lower, they develop into one sex, male or female, and if it's higher, they develop into the other. I can't remember off the top of my head whether lower temperatures give rise to males and females, but it is a temperature-determined trait. And the parent, the female usually, can exercise some control over that by how often she goes into the water and then crawls up onto the nest to let water percolate down into the nest. So she can cool the nest down by going into the water more frequently and coming up and basically dripping onto the nest, or by essentially watering the nest less frequently. And there is some evidence that female alligators and crocodiles sort of subconsciously monitor the sex ratio of their environment. Are there too many males? Are there too many females? And will alter the temperature of the nest accordingly to produce the appropriate type of offspring. So reproductive biology is a fascinating field, and there's lots of interesting phenomena to explore. Humans just represent one little facet of sex and reproduction in nature. So, putting it all together, of course, when we talk about the male reproductive system, then of course we're going to be looking at a system that is designed to do two things. One, to produce sperm cells, the gametes, the smaller haploid motile gamete that we call sperm cells. And secondly, it's going to be designed to introduce those sperm cells into the female body. Because again, remember, humans perform internal fertilization. And so males will have a penis for introducing gametes into the female reproductive system. The female reproductive system is going to be designed to do three things. It's going to be designed to produce the female gametes, the egg cells or ova. It has to be designed to receive sperm from the male. And so there is a receptive organ called the vagina designed to receive the male penis for internal fertilization. And then finally, the third extra function that the female system has to perform is it has to support and nourish the developing fetus, because again, we have internal development. And that is, of course, going to be the uterus. So when we look at these two systems over the next 2 weeks, we want to keep in mind the functions that they have to serve. Production of gametes for both introducing sperm into the female body for the male, receiving sperm from the male for the female, and housing the developing fetus. Now, in both cases, the reproductive systems consist of what we call primary and secondary sex organs. The primary sex organs are the gonads themselves, which are the organs that produce the gametes. Of course, these are the testes in the male and the ovaries in the female. And then we have a suite of secondary sex characteristics, which are organisms other than the gonads that are involved in reproduction. And so this would involve, in males, for instance, the system of ducts and tubes that carry sperm out of the body, various glands that produce fluids that help nourish the sperm cells on their way to and into the female body. In the female, it would involve, again, some tubes to carry the egg cells, the ovulated egg cells, to their proper location in the uterus. The uterus itself involved in housing the fetus, and the vagina, which, as I said earlier, receives sperm from the male. External genitalia are those parts of the reproductive system, of course, that we can visibly see from the outside. And internal genitalia are generally those that are located up in the pelvic cavity and cannot be seen from the outside. The secondary sex characteristics are all the other differences between males and females that are influenced by usually the presence or absence of the different hormones that males and females have. Of course, males we associate with testosterone, and females we associate with estrogen. Now, there are some technically sex characteristics that both sexes have. So, for instance, pubic and axillary hair that grow in during adolescence when the sex hormones begin to rise. Those are characteristics that both sexes have. And of course, they're useful for, as we talked about in AMP1, for retaining scent produced or manufactured by bacteria in our hair follicles as a result of feeding on apocrine sweat. But then there are other secondary sex characteristics that are different between males and females. So males have a larger larynx with a deeper voice. They have facial hair, more hair, of course, on the torso and on the arms and legs. Generally, lower hypodermal fat content, stronger muscles, higher red blood cell counts, etc. So some things can be seen, some things are not seen. The general rationale or reason why males have these characteristics is, one, they equip males to compete with each other for mates. This is the traditional evolutionary view, is that males had to be stronger because there's more competition in ancestral conditions between males for mates than there was between females for mates. And also just to help with basic sex distinguishing male from female, so individuals can identify appropriate mating partners. Females have a different set of characteristics, largely driven by the absence of testosterone and the presence of estrogen. So body fat tends to be distributed more under the dermis and the hypodermis, and placed more around places like the hips and buttocks, for instance. Of course, females have permanently enlarged breasts or mammary glands, and relatively less hair, especially on the torso and the face. So these, again, all help us distinguish male versus female. The excess body fat probably also helps adapt a female for gestation, for carrying a fetus and having energy reserves to fuel that costly endeavor of carrying a fetus for 9 months. We've already talked about the X and Y chromosomes. if you think about it carefully, what you realize because of this XY sex determination is that the sex of a child is determined by the type of sperm that fertilizes the egg. Since females are XX in terms of their genotype, when they produce ova or egg cells, the only kind of sex chromosome ova can carry are X chromosomes. That's all. X chromosomes. That's all that's available from the mother. It's the father that can produce two different kinds of gametes. Half of the father's gametes produced will carry an X chromosome, and approximately half will carry a Y chromosome. And so therefore it's the type of sperm cell that fertilizes the egg that will determine the sex of the offspring. So an X-carrying sperm cell will produce a female offspring because then the zygote will be XX. A Y-carrying sperm cell will produce a male offspring because the genotype then of the zygote will be YX. And that, of course, is male. And here we see a visual of what I just talked about in terms of sex determination by the gamete. Now, initially, fetuses are undifferentiated. They're not really male or female. But at about 5 or 6 weeks, they start to develop the gonads. And the gonads develop up, as I may have mentioned earlier, near the kidneys. And so you'll notice we don't want to go into all this detail, but notice the word nephric here. Nephric, paramesonephric ducts, mesonephric ducts. These are tubes that are up near the kidneys and actually develop from the same embryonic tissue that gives rise to the kidneys. And so in terms of development, both ovaries and testes start their development up by the kidneys and then have to descend down from the kidneys to their final place in the adult or in the child. The difference that ultimately determines whether a neutral fetus will become male or female is a gene that's carried on the Y chromosome called the SRY gene. The SRY gene codes for a specific protein, and that protein, when present, will initiate the development of testes as opposed to ovaries. And so the testes then begin to secrete testosterone just at the beginning of the fetal stage, which occurs about 8 to 9 weeks. And that testosterone that's coming from those initial testes is going to dictate then male development from that point on. In females, what we have is essentially estrogen. Estrogen is high anyway in the fetus, both fetuses, because of the estrogen coming from the mother. And in the absence of the SRY gene, and in the absence of that Y chromosome, all fetuses will naturally develop and become female because of the presence of this estrogen from the mother's the mother's system. And so really female determination of the female state is not necessarily due to estrogen. It's mostly due to the absence of the male hormones because of the absence of the SRY gene on the Y chromosome. And so we see that in the males and females, there are similar patches of tissue that develop. They just become different things in the different sexes, again, based on the influences of testosterone, its presence or absence. So the same structure that becomes the head or glands of the penis is going to become the clitoris in the female. The folds that become the penis in the male will become the labia minora in the female, and the folds that become the scrotum in the male will become the labia majora in the female. So even though in the adult the anatomy looks very different, embryonically we really start with the same basic pieces, and under the influence of the sex hormones, those pieces just develop slightly differently to become the different male and female anatomy. By week 12 of development, the fetuses are either male or female, and they are distinct. And from that point on, of course, ultrasound can be used to determine the sex of the fetus. And here again we see just an example of how similar the two look before 8 weeks or so. But then by 12 weeks, the differences become very clear between male versus female. As I mentioned earlier, the gonads begin their development by the kidneys and migrate down the pelvic cavity. In the case of the ovaries, of course, they don't descend that far. They simply descend down into the pelvic cavity. In the male, of course, the testes have to descend not only down to the pelvic cavity, but then on through an inguinal canal, which is a passageway out of the body cavity and into their normal place out in the scrotum, the sac-like scrotum in which they sit. Sometimes this inguinal canal doesn't close up, doesn't seal up properly following testicular descent, and you can have a hernia in that location, a weakness in the body wall, where you might actually get a projection of pelvic organs into the scrotal sac. And this is the most common type of hernia that you find in males. It's referred to as an inguinal hernia. Now, you may have heard that in some cases one or both testes do not descend all the way down into the scrotum. This is referred to as cryptorchidism, or simply undescended testes. It does occur in about 3% of males. And what happens is when they're born, the testes, one or both, are not in the scrotum. They are either still up in the pelvic cavity or somewhere in the inguinal canal and haven't descended all the way. In many cases, the descent completes itself within the first year of infancy. If not, if the testes don't descend all the way into the scrotum after the first year or so, then some correction needs to take place. Sometimes it's simple testosterone injection to fuel the descent, or sometimes a surgery can be done to simply pull the testes down into the scrotum. If the testes are left up in the body cavity, that leads to sterility and can lead to an increased probability of testicular cancer. So it is important to get that addressed. Here we see a diagram again of the testes, which must descend down through this inguinal canal right here. Descend down the testis, again starts up actually in the abdominal cavity, and it's coming down through the pelvic cavity and then through this canal called the inguinal canal, right here, passing through there and down into its final position in the scrotum. The reason testes are in the scrotum, by the way, is because in order to function and produce sperm cells, they have to be slightly cooler than normal body temperature. And since this location outside of the body cavity provides them with that cooler temperature. And notice here the inguinal canal then should seal up and seal around the various blood vessels, nerves, and tubes that still have to pass up and down between pelvic cavity and scrotum. All right, so it's finally time for us to start looking at the male reproductive anatomy now that we have a good firm background on reproduction and sex. And so let's begin. Here we have a basic diagram of the system, and we're going to go through it bit by bit. We're going to start with the external genitalia, which are of course the scrotum and the penis. The scrotum, as we've already said, is a sac-like structure in which the two testes reside, and again they reside there because of the necessity for having a cooler temperature in order to be functional. This region, by the way, called the perineum, is everything that can be located between the pubic symphysis up front, between the two hip bones, down to essentially the ischial tuberosities of each hip bone, and then back to the coccyx. So this whole diamond-shaped region here is referred to as the perineum in both sexes. And here we see again a cadaver view of the scrotum. So it is, of course, a pouch, a sac-like structure consisting of skin. There are some muscles, sheets of muscle underneath the skin, and some fibrous connective tissue, of course, to help hold it all together. The skin is typical in that it has sebaceous glands, pubic hair, or hair that's similar to pubic hair, innervation, etc. Just typical qualities of skin. There is an internal septum, a wall inside that divides the sac into a right and left compartment, of course, containing the right and left testes. There is a seam, a medial seam on the surface of the scrotum called the raphe, and this marks the location of that medial septum. The spermatic cord is not technically an external genitalia structure, but we might as well talk about it while we're talking about the scrotum. The spermatic cord is a bundle of fibrous connective tissue that passes up or through the inguinal canal into the pelvic cavity. It passes through the inguinal canal. So it consists of essentially vessel-like structures that need to communicate between the pelvic cavity and the scrotum. So what does it contain? It contains blood vessels, of course, arteries and veins, lymphatic vessels, of course, always for drainage of interstitial fluid, nerves for innervation of the structures inside the scrotum, and then a tube called the ductus deferens, which, as we'll see, is going to carry sperm from the testes out of the body, eventually out of the body. So that whole structure is known as the spermatic cord, and again, it passes through the inguinal canal, and there are rings of connective tissue that surround it and hold it in place. And again, we already referenced that the testes are in the scrotum because of the need for cooler environment. They have to be about 2 degrees Celsius cooler than typical body temperature. And here again we see the scrotum, the testes down here in the scrotum. There's the septum separating the scrotum into 2 compartments, and the spermatic cord, which leads up and away from the testis through the inguinal canal up into the pelvic cavity. Here we see another view of a testis in the scrotum, again with the spermatic cord. This time the spermatic cord's been exposed, been opened up, so you can see the various blood vessels, nerves, ducts, tubes that are located within it. And of course the entire structure is surrounded by connective tissue to hold it in place. A diagram instead of a cadaver photo of what we've been talking about. Again, the scrotum with its median septum separating it into 2 compartments, the testes in each compartment, and the spermatic cord containing vessels, nerves, ducts, and so on, leading up through the inguinal canal. So now it's time to talk about the primary organs of the male system, the gonads themselves, which are the testes or testicles. The testes are both endocrine and exocrine glands. For their endocrine function, of course, they produce testosterone, the sex hormone, primarily. For their exocrine function, they produce sperm cells. And you may recall way back in A&P1, we said there were certain types of glands called cytogenic glands that were glands that had as their secretion entire cells. And the only 2 cytogenic glands in the body were the testes and the ovaries. So technically, sperm cells are a secretion of an exocrine gland, but it's not a molecule, it's an entire cell. This is, again, what we call a cytogenic type exocrine gland. Each testis is oval, and about 4 centimeters long, so a little less than 2 inches long, and a little, just about an inch in diameter. And there are a couple of connective tissue layers that we want to appreciate associated with each testis. First of all, the outermost covering of a testis is something referred to as the tunica albuginea. Tunica albuginea. Well, the outermost actually is the tunica vaginalis, and then on the surface of the testis itself is a tunica albuginea. And this consists essentially of fibrous connective tissue, collagenous fibers, and the tunica albuginea will send sheets or walls, septa, diving down into the testis interior. These are referred to as septa, testicular septa, and this divides the testis up into lobules. There are quite a few of these septa in each testis, so each testis actually gets divided up into 250 to 300 little wedge-shaped lobules. And again, that's created by these connective tissue extensions of the tunica albuginea down into the internal testis itself. So here again we see a testis sitting in the scrotum in a cadaver. Here we see a cross-section through a testis, and what you can see is in white you see that tunica albuginea. Albu generally means white, so it's usually a whitish layer, and it extends little, again, sheet-like extensions of itself called septa into the testis, and you see highlighted in green various septa, various sheets here. And then, of course, all the material between the septa, as we said, is referred to as the lobules of the testis. Within the lobules, you have duct-like structures known as seminiferous tubules, microscopically small tubes that are located within the lobules. You generally have 1 to 3 seminiferous tubules in each lobule. Each seminiferous tubule is lined with a special epithelium of germ cells, and it's these germ cells that line the seminiferous tubules that produce the sperm cells. In addition to the seminiferous tubules, we also have a group of cells known as nurse cells. And these nurse cells are there to basically fill in the space between the germinal cells, the cells that are actually involved in making the sperm cells, just to fill in space between them and also to metabolically support them. So you see here, almost like glial cells in a way. I was thinking how the germ cells have to physically support the germ cells and metabolically regulate the environment of the germ cells, right, in terms of nutrients, waste removal, and other needs. So again, we have highly specialized cells, just like neurons are in the brain, and they need to be taken care of. Glial cells take care of our neurons. Nurse cells take care of our germinal cells in the seminiferous tubules. We'll come back to the seminiferous tubule in just a few moments and take a look at this structure. Now, again, these seminiferous tubules are coiled up inside each lobule of the testis, but between the coils, we do have cells sandwiched between separate coils of seminiferous tubules, and those are referred to as interstitial cells. So cells that are in the testis, in the lobules, but not part of the tubules, in between the tubules, are known as interstitial cells. And these are important because these are the endocrine component of the testis, and these are the tubules, these are the cells that produce testosterone. So the seminiferous tubule does not produce testosterone. It produces sperm cells. It's the interstitial cells between the seminiferous tubules that make testosterone and secreted into the bloodstream. So here again we see the seminiferous tubule under the microscope, and again it just looks pretty much like a typical tubule. the thing that's most noticeable about the seminiferous tubule is that if you look at the center, you can see, if you look very carefully, sperm cells. You see the little sperm cells with their heads and then long flagella, a very characteristic looking cell, and that's one of the best ways to note that you're looking at a seminiferous tubule. And so, to make a long story short, for now, just to preview, your germinal cells, your stem cells, are located here at the edge of the seminiferous tubule, and those are the ones that will initiate meiosis, a type of cell division that cuts the number of chromosomes in half. And as meiosis progresses, the daughter cells will get pushed downward towards the center of the tube. By the time you get to the end, you've got the 4 cells, the 4 products of meiosis, 4 haploid cells. And then they start to grow tails, flagellae, and by the time they make it to the luminal surface, they are almost done with their development, and so they simply get released. And as they're released, of course, they accumulate in the center of the seminiferous tubule. So we've got meiosis basically occurring from the outer edge towards the center of the seminiferous tubule, and the byproducts of meiosis are what are released, the sperm cells. Again, scattered between these obvious cells that are in various phases of meiosis, you do have the occasional nurse cell. Another name of the nurse cell, by the way, is the sustentacular cell. And you can see here in the highlighted nurse cells, they are scattered around in between the cells that are involved in meiosis at this point. There is something called a blood-testis barrier, somewhat similar to our blood-brain barrier. So the nurse cells that, again, support and surround these developing sperm cells, they form tight junctions between each other. And remember, tight junctions are junctions between cells that are fairly impermeable. They do not allow things to pass through very well. And so the nurse cells essentially all hook up with one another and create a barrier between the blood and the sperm cells that are developing in the seminiferous tubules. This is important because germ cells, gametes, are immunologically different from normal body cells. They're haploid cells. And so the molecules that they express at their surfaces are not the same as what you would see in a typical diploid body cell. And so they would be attacked by our immune system as foreign bodies. And so to prevent the immune system from being able to attack the developing sperm cells, we need to isolate them from the bloodstream so lymphocytes, macrophages, things like that cannot get to them. And so that's the basis of this blood-testis barrier. It really is to prevent our own immune system from attacking our developing sperm cells. Once again, the histology of the seminiferous tubules. Once again, you see a focus on the seminiferous tubule here, and we're going to have meiosis occurring, eventually releasing sperm cells into the lumen of the tubule. And again, we're going to talk about these stages of meiosis here in just a few minutes. I did want to point out or remind you that between neighboring seminiferous tubules, portions of seminiferous tubules, you do have masses of cells that are not part of the tubes. They just kind of help fill up the space between the tubes. These are referred to as the interstitial cells. And these are the cells that manufacture testosterone, those interstitial cells right there. So we're doing a little histology as we go. Now, the seminiferous tubules will all ultimately lead to a rete testis, which is a network of tubules on the backside of a testis. And the rete testis really serves as a collection site for sperm from the seminiferous tubules. So the sperm tend to get washed out of the lumina of the seminiferous tubules by fluid that's secreted by the nurse cells, so that creates an initial flow. The sperm are not mature enough yet to swim on their own, so they have to be washed towards the rete testis by this fluid coming from the nurse cells. We have a couple of slides here talking about the blood supply to the testes. We just don't have time to go into the countercurrent exchange mechanisms which help cool that blood. And I also want to remind you that, again, we do have nerves that pass through the spermatic cord down into the scrotum as well to innervate the testes and the tissues of the scrotum. All right, from the rete testis, then the sperm will flow into what are called efferent ductules. Efferent ductules, you only have a few of these. Remember, you have hundreds of lobules that feed into the rete testis, and then the rete testis will then trim it down and feed into only about 12 or so what are called efferent ductules. These are still small, but notice they're ciliated. And so now we've got cilia helping to create a current that's going to, again, draw that fluid out of the rete testis and transport it on to the next region of the male reproductive system, which is the epididymis. So the efferent ductules are about 12 small ducts that use cilia to create a flow to move sperm from the rete testis to a storage site known as the epididymis. The epididymis sits on the top and back side of each testis. So there are two, one for each testis. And again, these are primarily sites of sperm maturation. So sperm finish their development, maturation here, and storage. And sperm, by the way, can be stored for up to 2 months and remain fertile. After that, they start to die and fertility drops. The epididymis essentially is a single tube, so the 12 efferent ductules eventually fuse together to form the single tube, which we call the epididymis. It is highly coiled, folding back and forth on itself, and so ultimately it's actually 6 meters long, which is amazing, but it's packed into a very tiny space within the each compartment on each side of the scrotum. The sperm, again, finish maturing here in the epididymis. By the time they're mature, of course, they're capable of fully independent swimming. If the sperm cells are not ejaculated and they are stored in the epididymis for 60 days or more, as I said, they begin to die at that point and they disintegrate, they break down, and the epididymis will reabsorb their contents and recycle them, and use that energy and resources to manufacture new sperm cells. So here again we see a picture of a cadaver testis, and what you see sitting on top of and then behind would be the epididymis. So this structure right here that seems to cap the testis, that is the epididymis. Another view of the epididymis, highlighting its various parts. We don't have to worry about the head or the body or the tail. We just need to know epididymis. The epididymis then, again, is the site of storage. When it's time to release sperm for internal fertilization of the female, the sperm cells are then moved along a route that will take them out of the body. And the main tube that carries the sperm cells away from the epididymis is known as the ductus deferens, also known as the vas deferens. This is a highly muscular tube, which is about 45 centimeters long. To put that in context, 45 centimeters is about 18 inches or so, somewhere in that ballpark. What's interesting is, in order to pass out of the body through the vas deferens or ductus deferens, the sperm cells have to pass up from the scrotum through the inguinal canal as part of the spermatic cord, up into the pelvic cavity, where the ductus deferens kind of curves around the backside of the urinary bladder and then over the top. And then eventually from the top of the urinary bladder, it's going to ultimately fuse with the urethra, and then the sperm will be able to come out of the body that way. So the sperm take a fairly circuitous route to get out of the body. They go from the testes and the scrotum up into the body cavity, and then they come back out. And so the ductus deferens, as we said, continues up. On the back side of the bladder, it widens into a region known as the ampulla. And technically, the ampulla, this wide portion of the ductus deferens on the back side of the bladder, the top back of the bladder, that is technically the end of the ductus deferens. Because what happens after the ampulla, or at the ampulla, is that a duct coming from an accessory gland will fuse with the ductus deferens. And once the ductus deferens fuses with this duct, it becomes something else. So what organ has a duct that leads or fuses with the ductus deferens on the top back side of the bladder? It is known as the seminal vesicle. And the seminal vesicle provides the principal, or the greatest amount of what we call seminal fluid, in which the sperm cells will be immersed from this point forward. Once the ductus deferens and the duct of the seminal vesicle, once the ampulla, in other words, fuses with the duct of the seminal vesicle, they then become a very short tube known as the ejaculatory duct. The ejaculatory duct, as I said, is very short, less than an inch, and it runs from that top back portion of the bladder, or that, I shouldn't say top, that back side of the bladder, down through the prostate gland, and it eventually fuses with the urethra. So it would really help to see a picture of all this. So let's look at some pictures here. So again, the testis with its tunica albuginea right on the surface, this white fibrous tissue which sends septa, shown in blue here, these sheets of connective tissue down, separating the testis up into discrete lobules. Again, 250 to 300 of these lobules per testis. Inside each lobule is 1 to 3 highly coiled tubes known as seminiferous tubules, and that again is where the sperm cells are made. In between the tubes of the seminiferous tubules, you'll also have interstitial cells in here, which you can't see. Remember, those are making testosterone. What we didn't see earlier, or reference earlier, was what I said was the outermost tunica vaginalis. The tunica vaginalis is another layer of connective tissue that's found just outside the tunica albuginea. And the tunica vaginalis is actually a double-layered sheet of connective tissue. And you'll notice there's a space. There's actually a space between the two parts of the tunica vaginalis. This is a fluid-filled space that helps cushion and protect the testis that essentially floats in the fluid in the tunica vaginalis. And you'll also notice the outer layer of the tunica vaginalis is continuous with the connective tissue fascia that surrounds the spermatic cord. All right, on the back side of the testis, remember we said there was the rete testis, which is collecting sperm that's coming from each of the lobules, the seminiferous tubules in each of the lobules. And again, at this point, it's the fluid manufactured by the nurse cells, or the sustentacular cells, that is propelling the sperm towards the rete testis. Then we have efferent ductules, about 12 efferent ductules that are going to, by the action of cilia, draw the fluid out of the rete testis into the epididymis, which is this where the sperm cells finish their maturation and can be stored up to 60 days. This looks like it's many coiled tubules, but it's actually a single tubule that's coiled over and on top of itself many times. When it is time to release sperm from the male body, the sperm will get propelled into the ductus deferens, or vas deferens, which is a single tube that leads away from the tail of the epididymis and travels up into the spermatic cord, where eventually it passes up again into the pelvic cavity of the body. Here we see a cadaver picture of the ductus deferens traveling up through the spermatic cord. As you are probably aware, a very effective way to sterilize males, to voluntarily sterilize males, is to simply sever the vas deferens. If I sever the vas deferens, then there's no way for sperm cells to take this route back up and then out of the body. So by effectively blocking the pathway out of the scrotum, you sterilize the male. This, of course, is referred to as a vasectomy, literally meaning a cutting of the vas. Here we see a ductus deferens under the microscope in cross-section, and you notice here in blue the tremendous thickness of the smooth muscle in the wall of the ductus deferens. And it does have a longitudinal and a circular layer, so very much like intestines, for instance, it's capable of peristaltic waves of contraction, and this is what's going to propel the sperm from the epididymis up the ductus deferens up into the pelvic cavity. Again, going back to this slide, I should also mention that the male urethra has distinct regions, distinct portions to it. And so this initial part of the urethra that comes out of the bladder, that is referred to as the prostatic urethra. This is known as the prostatic urethra. And then after it fuses with the ejaculatory duct, it becomes the membranous urethra. For a very short period of time, this is referred to as the membranous urethra following this fusion. And once we enter the penis, then it is referred to as the penile urethra. So in this region, it is called the prostatic urethra, and this is the part that's got pores in it that receive prostatic fluid. Then we have the membranous urethra, this short section here. And then from about this point on, we have what's called the penile urethra. All right, and so the prostate gland produces about 30%. Now, the prostatic fluid makes up about 30% of the semen's volume. And again, towards the end of the presentation, we'll come back and talk about the contents of the prostatic fluid. And here again, we see the seminal vesicle in a cadaver located at the back bottom of the bladder. I was saying back top of the bladder earlier, but I was mistaken. I meant the posterior bottom part, so it can pass into the prostate gland directly, right? So that ejaculatory duct can travel directly into the prostate gland, which is again below the bladder. And then, of course, the third gland, as we said, is the bulbourethral gland. Sits below the prostate, and the bulbourethral gland produces about 5% or so of the semen, and it produces a clear lubricating fluid that helps lubricate the head of the penis during copulation. And again, we'll come back to the secretions of these three glands in a little more detail towards the end of the presentation. All right, so once again, here we see a different view of all of the components of the system, the testes with the epididymis leading to a vas deferens, comes around to the backside of the bladder, ampulla, enlarged ampulla, fuses with the duct coming from the seminal vesicles on each side. At that point, you have the very short ejaculatory duct, which then fuses with the prostatic urethra, just beneath the bladder where the prostate gland is. You can see from this anatomy why enlarged prostates often cause difficulty in males with urination. Enlarged prostates will compress the urethra and make it difficult to initiate urination in some cases. The enlarged prostate can also press up from beneath on the bladder, reducing the bladder's capacity, somewhat reminiscent of how a fetus might press on a female bladder from above. In either cases, by pressing on it, we're reducing its capacity and increasing the frequency of urination. The prostatic urethra, again, will pass down into the membranous urethra, at which point we have the bulbourethral glands. And once again, I want to point out that I said earlier that the bulbourethral gland was a single gland. And once again, I don't know what I was thinking, because I know full well that it's there's two of them, one on each side. So it's only the prostate that's a single gland. So I apologize for a couple of errors that I've made so far on this. The membranous urethra then becomes the penile urethra within the penis. As we know, the prostate gland, I mentioned that it can enlarge. Sometimes this is cancerous, sometimes not. If it's non-cancerous, this is referred to as benign prostatic hyperplasia. And as I mentioned, it compresses the urethra and interferes with flow. And this can result in bladder and kidney infections as the urine backs up and doesn't exit those organs as rapidly as it should. Sometimes enlargement of the prostate gland indicates a prostate cancer. Prostate cancer is the second most common cancer in men after lung cancer. In fact, in both sexes, lung cancer is the #1 cancer. In females, the second most common cancer is breast cancer. In males, the second most common cancer is prostate cancer. We hear a lot more about breast cancer than we do about prostate cancer, and the reason probably is not because it's less common, but it's less fatal. The prostate cancer can go unnoticed for many, many years. It's a very slowly developing cancer, and so it oftentimes with regular checkups can be detected and treated, and doesn't have the same mortality rate that breast cancer has in women. If it is allowed to progress without treatment, of course, like any cancer, it can metastasize away from the prostate gland to other organs and can become very serious, obviously. And so the way we detect it is through a couple of different exams. There's the digital rectal exam that is performed regularly during regular checkups, where the prostate gland is palpated through the rectal wall. This is the easiest way to get close to the prostate gland, is through the rectum, and a physician can feel whether the prostate has a normal size to it or not. And then if it is enlarged, of course, to detect whether it's a cancerous condition or not, you look for levels of certain chemicals, in this case serine protease, which is known as PSA. That's probably the most familiar test for prostate issues, serine protease. Also acid phosphatase, both of which are released from cancerous prostate tissue. All right, from here we move on to the penis. Of course, the penis is what we call the intromittent organ of the male reproductive system, meaning this is the part designed to be inserted into the female reproductive tract to facilitate internal fertilization. About half of the penis is internal, and the other half is externally visible as the shaft and the glands. The glans penis is the almond-shaped tip of the penis and the most sensitive part of the penis that responds to stimulation. The shaft contains essentially erectile tissue, which is essentially spongy vascular tissue that can fill with blood, causing the penis to go from a flaccid condition to an erect condition, which is necessary for insertion into the female reproductive tract. The skin over the shaft is relatively loose to allow for that expansion, and the skin also extends over the glands to protect the glands and maintain sensitivity. This portion of the skin that extends over the gland is known as the prepuce of the penis, also known as the foreskin. Now, in American culture, it's been very traditional to perform circumcision, which is the removal of the prepuce to expose the glands of the penis. This is done shortly after birth, and it got started as a practice in America because of a study that was done indicating that uncircumcised penises led to a higher incidence of penile cancer. And this is a study that was done by the Navy, I believe, back in the 1940s. I'm not sure exactly on the decade, but somewhere back then. Subsequently, it was discovered that there were many problems with this particular data set. And at this point, we don't really believe that circumcision makes any difference whatsoever with regard to incidence of penile cancer. And so at this point, circumcision has become really a very elective kind of procedure. And I think you're finding more and more that in America, parents are choosing not to circumcise their offspring because they realize really there's no solid health-related reason to do it. It really has become just a cultural practice, and it's a fairly invasive, disruptive, painful procedure to perform for the sake of a cultural standard. Some people argue that circumcised penises are easier to take care of in terms of cleanliness. There is a secretion called smegma. It's a sebaceous secretion that's produced by sebaceous glands in the glans penis, and it can accumulate under the foreskin. But with proper hygiene, that really is not a problem. So there's really no good medical reason to perform circumcision, and it's just really up to the parents at this point whether they think the cultural pressure to do so justifies it. As I mentioned, the shaft of the penis consists of erectile tissue that fills with blood to cause the erection of the penis. This occurs in response to physical stimulation and arousal, and again, it prepares the penis for intromission, for inserting into the female reproductive tract. If you do a cross-section through a penis, you see there are actually 3 cylinders of spongy tissue in the penis itself. There are 2 dorsal cylinders and 1 ventral cylinder. The 2 dorsal cylinders are referred to as the corpora cavernosa. The corpora cavernosa. These are 2 spongy cylinders of tissue that are on the top side or dorsal side of the penis. There's a single cylinder of spongy tissue on the underside or ventral side of the penis known as the corpus spongiosum. And it's the corpus spongiosum that encloses the penile urethra. So the urethra is within this cylinder that we call the corpus spongiosum. And it's the corpus spongiosum that at its distal end becomes the glans penis. So the glans penis merges with the corpus spongiosum. And again, they contain many spongy hollow spaces called lacunae, and we do have trabeculae that are sheets of tissue that break up the spongy areas that fill with blood. So here we see a diagram, a sagittal section through the penis of a cadaver, not terribly revealing. The better view really is a cross-section through the penis. And here we see a cross-section through the penis, and what you can clearly see are the 3 cylinders of tissue. The 2 dorsal cylinders are the corpora cavernosa. Each one is called a corpus cavernosum. And then the single cylinder below those, ventral to that, the corpus spongiosum, and at the center there you can see the urethra. Diagrammatically, again, it looks like this. The 2 corpora cavernosa dorsally there is a sheet, a fiber sheet between them called the septum, and then ventrally the, again, corpus spongiosum with the penile urethra at the center. Now, in terms of hormonal regulation, we're just going to talk about this briefly. You see on the second page of the list of terms, we have, at the very end, we have endocrinology, and we list testosterone, luteinizing hormone, and follicle-stimulating hormone. So there is a set of feedback relationships that regulate the hormone levels in males. And the best way to summarize it is to look at this particular figure. So again, it all starts with luteinizing hormone and follicle stimulating hormone, which recall come from the anterior pituitary gland. Also remember that the anterior pituitary gland gets its messages from the hypothalamus. Remember, the hypothalamus produces a releasing hormone that's going to stimulate the anterior pituitary to release luteinizing and follicle stimulating hormone. Luteinizing hormone specifically will travel to the seminiferous tubules or to the testes, and will specifically stimulate the interstitial cells. And remember what the interstitial cells do. The interstitial cells, of course, are the cells that make testosterone. So as luteinizing hormone increases, we're going to get an increase in the production of testosterone. One of the things that testosterone then is going to do is it will then turn right around and stimulate spermatogenesis in the seminiferous tubules. So higher levels of testosterone should lead to higher levels of spermatogenesis. FSH, on the other hand, follicle stimulating hormone, is going to travel to the nurse cells or the sustentacular cells and stimulate those cells to release something called androgen binding protein. Androgen binding protein. Androgen binding protein then to make a long story short, androgen binding protein is required if testosterone is to stimulate spermatogenesis. So when we said that testosterone stimulates spermatogenesis, it can only do that in the presence of androgen binding hormone. Well, androgen binding hormone again comes from the nurse cells, and they are stimulated to produce that by follicle stimulating hormone. So to get full function of the testes, both testosterone production and spermatogenesis, you need both luteinizing hormone and follicle stimulating hormone. Now, how do we regulate all this? Well, there are a variety of negative feedback mechanisms to prevent us from producing either too many sperm cells or too much testosterone. We'll start with testosterone here. Of course, we saw its effect is to stimulate spermatogenesis. Recall that testosterone also has other effects on the body, producing the secondary sex characteristics. Again, deeper voice, greater musculature, hairy torso, etc., beard. These are all the secondary sex characteristics, and the secondary sex organs during development. So development of a scrotum instead of labia, and a glans penis instead of a clitoris, etc. But in terms of the feedback mechanisms, the negative feedback mechanisms are shown in red here, where we have some inhibition. So as levels of testosterone rise, notice what's going to happen. The testosterone will feed back to both the hypothalamus and the anterior pituitary gland and have an inhibitory effect. In the hypothalamus, higher levels of testosterone that are too high are going to inhibit the hypothalamus, so it will produce less of its releasing hormone, which will then stimulate, which will result in reduced stimulation of the anterior pituitary gland. The higher than normal levels of testosterone will also directly affect the anterior pituitary gland and inhibit it as well. So through both these mechanisms, through both the hypothalamus and the pituitary, we're going to get an inhibition of the release of LH and FSH. And that then, as those levels go down, we're automatically then going to get a reduction in testosterone. And this, again, is a classic negative feedback loop that will keep testosterone levels essentially at a homeostatic level. So we don't see great fluctuations over short periods of time in testosterone like we're going to see in the female reproductive system, where we're going to see a very characteristic 28-day cycle of hormonal fluctuation when we look at that next week. The other negative feedback mechanism we have here is that as nurse cells are stimulated to produce androgen binding hormone, as they continue to get stimulated, they eventually build up and secrete a substance known as inhibin. Inhibin will enter the bloodstream and travel back to the anterior pituitary and specifically inhibit the release of FSH. And so as we get less FSH produced, that's going to produce less activity in the nurse cells, which means less androgen binding protein, which means reduced spermatogenesis. So this feedback mechanism is helping specifically to reduce the amount of sperm cells produced and keep that at a homeostatic level as well. So compared to the female system, as you'll see next week, this is a relatively simple set of hormonal interactions. Okay, and as we know, testosterone, we hear about this all the time in advertisements for various products on TV. Testosterone secretion declines with age. The peak levels of testosterone are somewhere at about age 20. And it declines markedly over the course of the next 60 years. And of course, as you lose testosterone, you're going to lose, you're going to have a reduction in the secondary sex characteristics, so reduction in muscle mass, sexual response, and so on. Andropause may occur. It doesn't occur in all males, but andropause may occur where males see a declining reproductive function and generally a decline in strength and healing rates and response to injuries and things like that, and athletic performance, etc., all associated with declining testosterone. And of course, sperm cells themselves decline as well. This is referred to as andropause, though again not as dramatic as the drop-off that we see in females, which is menopause, which we'll talk about next week. And even though sperm counts decline, it is possible for men to remain fertile and be able to father children throughout old age. And there have certainly been cases of men in their 70s and 80s fathering children. So again, unlike women, though male sexual function reduces, it doesn't disappear as it does in women following menopause. And so elderly men are capable of fathering children. In some men, they experience mood changes as the drop in testosterone triggers increases in FSH and LH. Remember, there's this negative feedback mechanism. So with less testosterone, we get more FSH and LH. And so some men notice this. Most men do not, but some men notice these surges of these two hormones, and it results in, again, traits that we often associate with menopause. But in andropause, you can get, again, mood changes, hot flashes, things like that. Perhaps the best known side effect of reduction in testosterone levels is erectile dysfunction, where the male is unable to produce an erection. And about 20% of men in their 60s suffer from erectile dysfunction, and this goes up to about 50% of men in their 80s. And again, you see advertisements all the time on in various media sources for treatments for erectile dysfunction. All right, the last topic we're going to cover, just to finish everything that we've been talking about, is the process of spermatogenesis, how it takes place in the seminiferous tubules, and then the contents of the semen, the fluid that the sperm are bathed in that comes from the accessory glands. So spermatogenesis is the production of sperm cells in the seminiferous tubules. And again, it involves germ cells, which are essentially stem cells, which are capable of then leading to a meiotic division, and that meiotic division then is going to produce daughter cells, which become the sperm cells. So what we're talking about here is meiosis, of course, and you just have to remember some basics of meiosis from intro bio. Goes through some of the same phases as mitosis, prophase, metaphase, anaphase, telophase, but there's two stages of meiosis. The first stage of meiosis is where we cut the number of chromosomes in half, and then the second stage, or meiosis 2, simply involves converting the chromosomes from dyad form to monad form, so we have single copy DNA in all the daughter cells. So take home thing to remember is for every one cell that begins meiosis, you end up with 4 daughter cells that have half the number of chromosomes that the original cell started with. The other important thing to remember about meiosis is because of the crossing over and recombination that takes place as pieces of chromatids break and get exchanged with one another, we get lots of variation produced as a result of meiosis. So none of the 4 daughter cells are identical to each other genetically that are produced in meiosis. They are all genetically distinct. So in addition to the mixing of genetic material that happens with sex, the process of meiosis itself is also critically important in producing the genetic variation that's important to the long-term survival of a species in terms of responding to environmental changes. So the meiosis is not the only kind of cell division that takes place in the seminiferous tubules. For instance, if every stem cell of the seminiferous tubule entered meiosis, you would quickly exhaust all the stem cells and you would just produce haploid sperm cells, and then you'd be done and you would never be able to do it again. So in order to maintain a population of stem cells throughout the life of the male, we do have to have some mitosis that's occurring too, some replacement. And so what happens is that the cells, the stem cells that are at the edge of the seminiferous tubule, and this is just a review of meiosis again, which we don't need this level of detail. So let's pass through all this detail on meiosis. I left it here for you in case somebody wanted to review it, but I'm going to assume that you remember the basics of meiosis. But I want to get back to the mitosis that begins everything. So in these seminiferous tubules, you have stem cells known as spermatogonia. And these stem cells, again, are right at the very outer edge of the seminiferous tubules. These spermatogonia divide by mitosis. Because they divide by mitosis, you're never going to run out of them. You will have these stem cell spermatogonia throughout the life of the male. So as a result of each mitotic division, and remember, from mitosis you produce 2 identical daughter cells. One of these identical daughter cells is going to remain at the periphery as a stem cell. So this is why we never run out of stem cells in males, because every time we do mitosis, one of the daughter cells persists as a stem cell. We call these type A spermatogonia. The other daughter cell of mitosis actually moves away from the outer edge of the seminiferous tubule and starts migrating towards the lumen, towards the center. This is a type B spermatogonium, and this is the one that's fated to undergo meiosis and ultimately produce the sperm cells. So mitosis is happening at the periphery, at the edge, to maintain always at least one type A spermatogonium, so you never run out. The type B spermatogonia get pushed towards the lumen and begin to undergo meiosis. So again, the type B spermatogonium enlarges and becomes a primary spermatocyte. Once its fate is set and it's no longer a stem cell, once it is now fated to undergo meiosis, it enlarges and becomes what we call a primary spermatocyte. This primary spermatocyte will undergo the first meiotic division, giving rise to 2 haploid cells, because remember, the first meiotic division is the reduction division. So this is where we cut things in half. At this point, they are referred to as secondary spermatocytes after the first meiotic division. Each secondary spermatocyte then will undergo the second meiotic division, and as this is happening, by the way, they're constantly migrating or moving towards the lumen or center of the seminiferous tubule. Each secondary spermatocyte will undergo the second meiotic division and result in two daughter cells known as spermatids. So by the time we call them spermatids, they have completed the two meiotic divisions, and now you have full-fledged haploid cells. So from a single primary spermatocyte, you're going to get a total of 4 spermatids. Correct? Because again, you've got 2 cells doing the second meiotic division. Spermiogenesis is the name we give to the differentiation or transformation of these spermatids into what we eventually call spermatozoa. And spermatozoa, by the time they become spermatozoa, they have sprouted flagella, and even though they're not actively swimming, they have all the features of the adult sperm cell. They finish maturation, remember, in the epididymis, and that's where they become motile. As we said earlier, as the spermatocytes undergo meiosis, they produce genetically distinct haploid cells that need to be protected from the immune system. And remember, I said that the nurse cells with their tight junctions form that blood-testis barrier that protects the developing sperm cells from the immune system. Again, another name for nurse cell is sustentacular cell, and yet an older name for nurse cell is Sertoli cell. So you may hear any of these terms used for these cells that protect the spermatids and spermatocytes from the immune system. Nurse cell, sustentacular cell, or Sertoli cell. Here we see a diagram of what we've been talking about. Always good to see a diagram. Here we're looking at a seminiferous tubule. Remember, interstitial cells would occupy these approximately triangular spaces between seminiferous tubules. That's where testosterone is made again. But we're going to look at this little wedge, this little section through the seminiferous tubule over here. So at the top is the lumen, the hollow center of the tubule. At the edge down here is the periphery. Again, you have your spermatogonia, your basically your stem cells here at the edge. They undergo mitosis to produce type A spermatogonia and type B spermatogonia. A type A spermatogonium is going to persist at the edge as a stem cell. The type B spermatogonium is going to start migrating down through the wall of the seminiferous tubule. It prepares for meiosis, and once its fate is set, it becomes what we call a spermatocyte. The spermatocyte undergoes the first meiotic division to produce secondary spermatocytes. Each secondary spermatocyte is going to undergo the second meiotic division, so from a single primary spermatocyte we end up with 4 spermatids. Notice that the spermatids do not have flagella, and so we experience then what's called spermiogenesis, where the spermatids alter their shape and start to take on the characteristic shape of sperm cells with a head of cytoplasm and then a long flagellum, not swimming yet, but these then are released into the lumen, where they will then be propelled by fluid from the sustentacular cells towards the rete testis. Speaking of the sustentacular cells, where are they? Well, they are all this beige type background material. All of this is happening within the confines of the sustentacular or nurse cells. So here we see one nurse cell. Right in the middle here, we see a second nurse cell. Off to the right here, we see a third nurse cell. These lines here represent the tight junctions between nurse cells. And so these meiotic divisions and development are taking place in the little spaces between nurse cells. And what eventually they're going to migrate towards the end during spermiogenesis. The spermatids actually enter the nurse cells and migrate through them and are released by the nurse cells almost in a kind of exocytosis, a weird kind of exocytosis of a whole cell. So remember, this is that unusual cytogenic type gland where the secretion from a cell is actually another whole cell. So a very unusual condition. And here we see an electron micrograph through the seminiferous tubules. And again, what's most obvious is the accumulation of flagellated sperm cells in the lumen of the tubule. And here we see spermiogenesis, the conversion basically of a spermatid to a spermatozoa, where you shed some cytoplasm and go through some other developmental processes that we would talk more about in lecture. So I'm not going to spend too much time on the spermatozoan. It is just simply the flagellated sperm cell. All right, the last topic, as I said, is the contents of the semen or seminal fluid. This is the fluid that the sperm cells are swimming in, and it's this fluid that is expelled during the male orgasm. As we said earlier, about 60% of the fluid comes from the seminal vesicle, about 30% comes from the prostate, about 10% of it is sperm and spermatic duct secretions that include the bulbourethral gland as well. Typical sperm count is as many as 50 to 120 million sperm cells per milliliter of seminal fluid. If you have less than that, or less than 20 to 25, that's considered infertility. So what that tells you is to be infertile and still have as many as 20 million sperm cells per milliliter tells you that not many sperm cells successfully make it to fertilize an egg cell. So you can have 20 to 25 million per milliliter of seminal fluid and still be considered infertile. As we'll talk about when we get to the female reproductive tract, it's a very inhospitable place for sperm. They just don't do well once they're inside the female reproductive tract. And they have to swim a fairly long distance from the perspective of a microscopic cell in order to even reach the egg cell. It has been argued that this is essentially an obstacle course for sperm cells that the female body intentionally sets up to ensure that only the best, strongest sperm actually make it to the egg cell. And so the vast majority of sperm cells that are released into the female reproductive tract do not make it far enough to fertilize the egg. And so that's why you can be infertile even though you still are making active sperm cells. So again, the prostate gland, as we said, produces a milky white fluid called prostatic fluid. It contains a variety of substances: calcium, citrate, phosphate. Some of these are involved in energy, right? Citrate provides energy, phosphate for ATP production. It does contain a clotting enzyme, which tends to cause the semen, once released, to bind up a little bit, to become semi-fluid or semi-solid. This is important in not allowing the semen to drip back out of the female reproductive tract once it's been introduced. Of course, it can't remain in that solid state. The sperm cells wouldn't be able to swim anywhere. So after a short while, that will get broken down and it will turn fluid again so that the sperm can swim. But initially, it clots it up to help keep it within the female body. The seminal vesicles contribute a yellowish fluid to the semen. And primarily the seminal vesicles provide energy sources for the sperm cells. So fructose, citrate again, prostaglandins, mostly fuel sources though for the sperm cells. It also contains a protein that digests the clotting enzyme of the prostate, as I said, which will then help liquefy the semen again and help the sperm swim up the female reproductive tract. So again, just going back to what we talked about, the clotting enzyme from the prostate activates a substance in the seminal fluid called proseminogelin, and it's actually the seminogelin that creates the clotting. And so the enzyme from the prostate does what enzymes usually do. It catalyzes a reaction, turning proseminogelin into seminogelin, which creates the sticky mass. And as I said, this helps it stick to the female reproductive tract and keeps the fluid from draining back out of the system. As I said, about 20 to 30 minutes later after ejaculation, another enzyme called serine protease from the prostate will break down the seminogelin, reliquefying the semen and allowing the sperm cells again then to start swimming. Now, what do sperm cells need to actively swim? Well, they need essentially two things. They need an energy source to provide energy for that active flagellar movement, and they need an elevated pH, a slightly alkaline pH. The pH issue is covered through prostatic fluid because prostatic fluid contains a buffer that buffers vaginal acidity. Remember I said just a few minutes ago that the female reproductive tract is inhospitable to sperm cells, and one of the ways in which it's inhospitable is the vaginal canal is fairly acidic, normally exhibiting a pH of about 3.5. But the prostatic fluid helps out with that because it buffers the vagina to a pH of about 7.5, which is perfect for the sperm cells to survive and actively swim. We also said, again, that they require an energy source for all this active swimming, and both the prostatic fluid and the seminal vesicles provide fructose and citrate and things like that, which provide basic energy for aerobic respiration and therefore ATP for the sperm cells. The sperm cells not only have to crawl up the wall of the vagina, they have to also pass up into the uterus, and to get into the uterus, of course, they have to migrate through the cervix of the uterus, the bottom entry to the uterus. The cervix usually has a mucus plug in it, and so that has to be thinned out or digested or dissolved for the sperm cells to pass through it. We have prostaglandins in the seminal fluid from the seminal vesicle that seem to be involved in thinning out that mucus so that sperm cells can swim through it. The prostaglandins, it's also hypothesized that these prostaglandins may stimulate contraction in the uterine wall especially, which may help spread the sperm throughout or the semen throughout the wall of the uterus. Now, the sperm cells can't just stop at the uterus. They have to continue swimming up the side walls of the uterus and enter the fallopian tubes. I know we haven't done much of any of the female reproductive system, but you may be familiar with these two tubes called fallopian tubes that arc out from the top of the uterus. At any rate, the sperm cells have to swim into the fallopian tubes, and if they make it that far, then they might encounter an egg cell coming from the ovaries, and if fertilization is to be successful and result in pregnancy, the fertilization event has to occur in the fallopian tube. So we see that we've got these microscopically tiny cells that have to swim, relatively speaking, a great distance against acidity and muscle contractions and mucus and make it up to the fallopian tubes. And by the way, there's cilia in the fallopian tubes that create a current against which the sperm cells have to swim as well. So it's a tough journey to get that far. That's why so many of the sperm cells don't make it that far. And so that's why, as we said earlier, you can have a sperm count of 25 million and still be considered infertile, because you might introduce that many sperm cells and have none of them make it as far as the fallopian tubes to fertilize the egg. So that's it for the male reproductive system. Next week we will cover the female reproductive system, and there is a second video to accompany this video. It's going to be much shorter than this video, and all it will be is a single model that we have of the male reproductive system, and I will show you the structures that we can see on that model. As for histology, it was embedded in this presentation once again, and so all the slides that we would normally have looked at, you have seen as part of this PowerPoint presentation. So I look forward to talking to you on the next video where we look at the male reproductive model.